Welcome to all students. Myself, O.P. Gozara, Assistant Professor, Seed Technology, Department Faculty of Agriculture and Veterinary Science, Prithvi Vidyapits, Women University, Jaipur. Today, we discuss about our topic, deteriorations of crop varieties. तो crop varieties है उसके अंदर जो deteriorations है वो किस तरीके से होता है and इसका जो maintenance है वो किस प्रकार से किया जाता है. Deteriorations of genetic purity. The genetic purity of a variety or trueness to its type deteriorates due to several factors during the production cycle. Scientist Kadam's 1942 mainly which gives different factors which responsible for deteriorations of varieties. Developments, variations, mechanical mixture, mutations, natural crossing, minor genetic variations, selection, influence of pest and disease, the techniques of the plant builders. Development variations in includes when seed crops are grown under environment with differing soil fertility, climate, photo periods, or at different elevations for several consecutive generations. Development variations may set as different growth responses. It is therefore prepared to grow the varieties of crops in the areas of their natural adaptation to minimize development shift. In mechanical mixers, the most important reasons for varietal deteriorations often take place at time of sowing is more than one variety is sown with the same seed trails. Throw volunteer plants of the same crops in the seed fields or throw different varieties grown in adjacent fields. Two varieties growing next to each other fields is usually mixed during harvesting and threshing operations. The threshing equipment is often contaminated with seeds of other varieties. Similarly, the gunny bags, seed bins, and elevators are also often contaminated, adding to the mechanical mixtures of varieties. Rogging the seed fields critically and using utmost care during seed productions and processing are necessary to avoid mechanical contaminations. Mutations in which do not seriously deteriorate varieties, it is often difficult to identify or detect minor mutations occurring naturally. Mutants such as Edwards in oats or rabbits ear in peas may be removed by loggings from seed plots to provide the seeds. Natural crossing, in which natural crossing can be important source of varietal deteriorations in sexually propagated crops. The extent of contamination depends on the magnitudes of natural cross fertilizations. The deterioration sets is due to natural crossing with undesirable types, disease plants or rock types. In self-fertilized crops, natural crossing is not a serious source of contaminations unless variety is well sterile and is grown in close proximity with other varieties. The natural crossing, however, can be major source of contaminations due to natural crossings are the breeding systems of the species, isolation distance, varietal mass, and pollinating agents. The directions of prevailing winds, the number of insects present, and their activity and mass of varieties. Also important considerations in contaminations for the natural crossings. The isolations of the seed crop is the most important factor in avoiding contaminations of the cross-fertilized crops. Minor genetic variations in this minor genetic variations can occur even in varieties appearing phenotypically uniform and homogeneous when they leaves. The variations may now lost during later production cycle, owing the selective eliminations by the natures. The yield trials of lines propagated from plants of breeder seed to maturations, the purity of self-pollinated crops varieties can overcome these minor variations. Due cares during the maintenance of nucleus and breeder seeds of crop, fertilized varieties of crop is necessary. Selected influence and of pest and disease. New crop varieties often are susceptible to newer race of pest and disease caused by obligate parasites and thus selectively influence deteriorations. The vegetatively propagate stocks also can deteriorate quickly is infected by virus, fungi, or bacteria. Seed productions under strict disease-free conditions is therefore essential. The techniques of plant breeders serious instabilities may occur in varieties owing to cytogenetic irregularities in the form of improper assessments in the release of new varieties. Premature release of varieties, still segregating for resistance and susceptibility to disease or other factors can cause significant deteriorations of varieties. This failure can be attributed to the variety testing programs. In addition to these factors, other heritable varieties variations due to recommendations and polyploidizations may also take place in varieties during seed productions, which can be avoided by periodically selections during maintenance of the seed stocks. Maintenance of genetic purity during seed productions, the various steps suggest mainly use of approved seed only in seed multiplications, inspections and approval of field prior to planting, field inspections and approval of growing crops at critical stage for verifications of genetic purity, detections of mixtures, weed and for freedoms from noxious weed and seed-borne disease, etc. Sampling and sealing of clean lots, growing of samples of potentially approved stock from comparisons with the authentic stocks. As like various steps suggested maintain genetic purity, providing adequate isolation to prevent contaminations by natural toxins or mechanical mixtures, roughing of seed fields prior to the stage at which they could contaminate the seed crops, periodic testing of varieties for genetic purity, avoiding genetic shift by growing crops in area in their adoption only. Certifications of seed crops to maintain genetic purity and quality of seeds, adopting the generation systems grow out test. Genetic purity maintenance in hybrid seed. Maintenance of the genetic purity of hybrid seed is a complicated one requiring elaborate procedures. Firstly, nuclear seeds of inbred lines. The nuclear seed of inbred lines can be maintained by cell pollinations, seed pollinations, or combination of two procedure hand pollination. Some breeder prefer sibling because it maintenance vigor. Selfing is used to stabilize inbred lines. In a change, breeding behavior is noticed. Some parent materials is preferably maintained by alternate selfing and sibling from one generation to other. Individually, sulfate or seabed years should be examined critically, discarding of types or inferior characteristics as like texture, color, seed size, chaff color, and shape of ear heads. The uniform ear are then threshed separately and planted in ear to row methods to easily detectors and discards of types from individual ears, if any. Alternatively, all the ears from an individual inbred lines may be composited for bulk planting in the next seasons. The hand pollination seed is sown on cleans, fertile soil having no previous crop of the same kind of variety during the previous year. It is rather important to ensure that the crop is well isolated with the requirement varying from crop to crop and depending upon the natures of the materials to be protected by isolations, the nature of the contaminants and the directions of the prevailing winds. The isolation can be achieved either by distance or by time. The inbred line may be composited for bulk planting in next seasons. Maintenance of genetic purity in inbred lines through hand pollinations and adequate isolations alone is not enough to achieve infections. The isolated field must be critically referred for oak types and other impure types prior to the setting of plants. The nucleus seed crop is harvested after physiological maturity. It artificial drying facility exits. Year two harvest lines are harvested separately and the plates. These are again critically examined of air characteristics, sorting out of all of colored disease or otherwise undesirable ears. If the overall percentage of type exceeds 0.1%, hand pollution should be repeated to produce the second year breeder seeds. The uniforms ears are bulked, dried in a clean dry bins, temperatures not exceeding 43 degrees centigrade, sealed, clean, treated with pesticides and stored under ideal storage conditions as breeder stock seeds. The seeds may be increased during the following seasons by paying adequate attention to isolations, probing etc. to maintain high genetic purity of the seeds. Nucleic seed of non-inbred lines to maintain in the genetic purity of the nucleus seeds of non-inbred lines. The number of plants for hand pollinations should be large enough to preserve genetic makeups of the variety. Narrow the genetic base by sieving only a few plants, about 5,000 plants or more. The civets here are examined critically, discarding of color, texture, or disease ones. Uniforms here are bulked, dried, shelled, cleansed, treated, and stored as usual. Other practice of seedling civets nucleus seeds are similar to those described earlier of for inbred lines. Rogging, however, need to be observed more critically by individuals with good knowledge of the material. The breeder stock seed thus produced from the nucleus seeds can be utilized to increase the breeder stocks of non-inbred lines, paying adequate attention to land requirements, isolation, probing, harvesting, and handling of seed to achieve maximum genetic purity. The breeder seed of the established varieties of cross-pollinated crops can be maintained by raising breeder seed crop in isolations and probing the crop thoroughly at various stages. It is often purified by mass selections. The crop is grown in isolations and fed carefully as described earlier. At maturity, about 20,000 to 25,000 to type plants are selected, harvested separately, and bulked after careful examinations. This constituents the breeder stock seeds. The seed may be carried over to ensure against possible failure or unforeseen shortage. सो मेनली प्रोडक्शंस जो डिटोरेशंस इन प्रो वेराइटीज है उसके अंदर मेनली जो आपका न्यूक्लियस सीड है उसका जो प्रोडक्शंस है वो मेनली इनब्रेड लाइन से और विथ नॉन इनब्रेड लाइन से किस तरीके से इसका जो प्रोडक्शन है वो किया जा सकता है तो इसके बारे में ये जो कंप्लीट स्टडी है मैंने आपको बताई है तो मेनली जो सीड प्रोडक्शन है वो आप किस तरीके से कर सकते हो या किस तरीके से नहीं कर सकते तो इसके लिए जो अपन को किस किस तरीके से सावधानियां वो रखनी चाहिए और जो सीड प्रोडक्शन है उसके लिए मेनली कई तरह की जो टेक्निक्स है या जो टेक्निकल पर्सन है उसको किस तरह का नॉलेज होना चाहिए तो उसके अकॉर्डिंग अपन क्या है कि जो सीड है उसकी जो जेनेटिक प्योरिटी है उसको मेंटेन रख सकते हैं और जेनेटिक प्योरिटी मेंटेन रखने के लिए जो फील्ड है उससे भी मेनली कई तरह की जो प्रॉब्लम्स आती है फर्स्टली उसको मेनली वहाँ से रिमूव करना पड़ता है और प्लस टेक्निकल जो पर्सन है उसको भी इसके बारे में कंप्लीट नॉलेज होना चाहिए कंप्लीट नॉलेज हो तभी मेनली जो अपना जेनेटिक प्योरिटी है जो या सीड प्रोडक्शन है वो बेटर हो सकता है इसके अलावा जैसे अपने गवर्नमेंट इंस्टीट्यूट्स है इंडियन काउंसिल ऑफ एग्री रिसर्च या जो एग्रीकल्चर यूनिवर्सिटीज है या नेशनल सीड कॉर्पोरेशन है वहाँ मेनली जो फार्मर है उसको मेनली ये जो चीज़ें हैं वो अवेलेबल हो सकती है एक फार्मर्स है उसको एक बेस्ट मेनली अगर सीड अवेलेबल होगा तो मेनली अपना जो सीड का प्रोडक्शन है वो बेटर रहेगा। तो मेनली जो पॉपुलेशन है प्रेजेंट टाइम में, जो पॉपुलेशन इंक्रीज हो रही है, तो उसके अकॉर्डिंग मेनली जो सीड है उसका जो प्रोडक्शन है वो इंक्रीज होना जरूरी है, क्योंकि पॉपुलेशन है उसकी फूड की रिक्वायरमेंट्स मेनली कंप्लीट करने के लिए जो सीड है उसके अंदर कुछ जो यील्ड है, सीड की जो यील्ड है उसको इंक्रीज करना जरूरी है। तो इसके लिए मेनली कुछ टेक्निक्स है वो ब्रीडर के द्वारा या जो टेक्निकल्स पर्सन है उसके द्वारा मेनली है जो न्यू क्रॉप वैरायटीज है उसको रिलीज किया जाता है। तो इसके लिए मेनली डिफरेंट फील्ड है उसको सिलेक्टेड किया जाता है। उस फील्ड के नज़दीक ऐसा कोई फील्ड नहीं होना चाहिए, प्लस उसमें जो कंटामिनेशन है वो नहीं आनी चाहिए। तो कई सारी जो मेनली प्रिकॉशंस है उसका यूज करते हुए मेनली जो जेनेटिक प्योरिटी है उसको मेंटेन किया जा सकता है। तभी मेनली अपना जो जेनेटिक प्योरिटी है वो सीड्स प्रोड्यूस हो सकता है। तो एज लाइक कि सीड प्रोडक्शन के लिए कई तरह की जो प्रोसीजर्स है उनको फॉलो किया जाता है। अदर जो मेनली सीड्स बेस्ड न्यूक्लियर सीड है उसका जो प्रोडक्शन है वो मेनली करने के लिए मेनली जो प्लांट है उसके बारे में कंप्लीट जो नॉलेज है उसके बारे में होना जरूरी है। अगर कंप्लीट स्टडी नहीं होती है तो कई तरह के जो टेक्निकल इश्यू है, टेक्निकल एरर है उसके द्वारा क्रिएट होती है। तो उस कंडीशंस में मेनली जो प्रोडक्शन है वो डिस्टर्ब होता है। तो इस कंडीशंस में मेनली ये आपके लिए एक हेल्पफुल हो सकता है कि जो अगर आप किसी कंपनी में जा रहे हो, सीट से सर्टिफिकेशन एजेंसी है वहाँ अगर आप जा रहे हो, या आप कोई भी रिसर्च कंपनी, रिसर्च एंड डेवलपमेंट उसमें जा रहे हो, तो वहाँ ये सारी जो टेक्निकल नॉलेज है वो आपको होना जरूरी है। ये नहीं होगा तो आपका जो शीट प्रोडक्शन है वो बेटर नहीं होगा, और आपकी जो क्रॉप वैरायटी है उसके अंदर कई तरह की जो प्रॉब्लम्स है वो क्रिएट हो जाएगी। ओके, थैंक यू स्टूडेंट्स।